This video is going to go over the phases of the cell cycle and mitosis. So this model right here represents a cell in interphase. Notice that there is a intact nuclear envelope, that the centrioles are not really doing anything, they're just hanging out over here to the side, and the DNA is in the chromatin form. All right. These two models represent prophase. This is early prophase and this is late prophase. You don't have to tell me early and late for the exam. What's happening during prophase is we're beginning to create the mitotic spindle. So my centrioles move to opposite sides of the cell and begin casting microtubules that will become the spindle. If we look in the nucleus, we notice that the DNA is starting to condense, and we're going to get rid of this nuclear envelope. So that by the time we're over here in late prophase, we have our DNA condensed, although it's not lined up yet, and we have our full mitotic spindle stretched across the cell without a nuclear envelope in the way. The next phase is metaphase, and in metaphase, each chromosome with its sister chromatid pair is lying across the center of the cell in what we call the metaphase or equatorial plate. This is easy to recognize because it's like a little straight line of X's through the middle of the cell. The next phase is called anaphase. And again, we have two models. We have an early anaphase and a late anaphase. The first thing that happens is the two sister chromatids let go of each other, and so we become two clusters of chromosomes. One is going to go to one pole of the spindle, the other one's going to go to the other pole of the spindle. So as soon as those sister chromatids let go and we can see this space between them, we call this anaphase. In later anaphase, the clusters of chromosomes have gotten closer to the poles. There's a larger space between them, and we have begun the other process called cytokinesis by forming this little dimple or divot in the side of the cell that is known as the cleavage furrow. The final stage of mitosis is telophase. And in telophase, the chromosomes have arrived at the two poles of the spindle. The DNA is now being resurrounded by a new nuclear membrane, and we can see remnants of the spindle stretching across between the cells, and we see a much deeper cleavage furrow as cytokinesis has continued. That is telophase. And then finally, we have the completed two daughter cells that have been completely separated through cytokinesis, encased in their own nuclei, and the DNA has relaxed back down into chromatin.